It's a great pleasure for me to introduce the next speaker. Actually, I know that Morten would really have loved to do it, but since he can't be here right now, I have to do it. So the next speaker definitely has had and has a great impact on the aging research field. Actually, I don't think he needs an introduction. He also has a great impact on this conference. One of the founders and co-organizers. It's a great pleasure, Alex, to have you here, and we are looking forward for your talk.
Thank you so much. It's a great privilege for me to be on the stage. First of all, I must thank everybody for coming, because in a position of a co-chair, it's a truly amazing event, one of the largest, I guess, in the world at this point in time. I'll go through my lecture, but after that, I'm also going to make a very important personal announcement. So I'll try to be brief during the lecture slides, but not to diminish the content. I'll go into detail into some of those. So today I'm going to talk about probably the most important journey that our company and myself took over the past 40 months. So currently we are kind of midway, phase 1. But I'm going to talk about the journey where we took a novel target all the way into human clinical trials using our AI platform, which took us about 5 years to build before we started doing any of our own research and development work in formulating our own pipeline. So all the legal disclaimers, not investment advice, no forward-looking statements. As a matter of fact, you won't be able to invest in Insilico in the near future. And a little bit about the company. So currently we're about 250 people, some maybe already more. We are very geographically spread. We span 9 time zones. And we also have over 80 contract research organizations that we work with. So we work with a lot of CROs, and we utilize what is called FTEs, so full-time equivalent scientists. So if you count them, we probably are over 500 now. This is the slide that everybody should know by heart, and I've presented it at our day 0 during Longevity Medicine Day. This is how drugs are discovered. Here you've got the different steps. Starts with target hypothesis. That's what most of the academics are working on. So very rarely the pharmaceutical companies discover their own targets. But when the target is discovered, and by the way, this is probability of success, this is time it takes to get to the market, this is the amount of money it costs in millions, usually on average for the industry. So target discovery is extremely expensive process, and most of the work is usually done by the academics, such as you. So once you identify the target, this is how long it's going to usually take to reach the market, and the probability of reaching the market is very low. Most of the failures in phase 2. So what we are trying to do is improve the probability of success by having multiple different AI systems working together in what we call the end-to-end approach. So we are an AI platform company, and we also have a pipeline of our own therapeutics. Currently, I have 31 therapeutic programs. So today I'm going to talk about one. So in 2020, we had to reinvent ourselves into this model where we have AI and drug discovery and drug development platform working together. Originally, we were just an AI company. In 2020, we made the decision to move from platform to a pipeline. And I got a co-CEO. So we actually have 2 CEO structure, Dr. Ren Feng, Harvard PhD, 11 years at GSK, and then he listed a company called Medicilin, took it all the way to $8 billion, and then joined me as a co-CEO of this organization. So now we have drug discovery veterans running the show. What I am responsible for and what I've been working on beginning of the company is the end-to-end software platform. So we can call it AI, we can call it software. It allows you to seamlessly identify novel targets. Many of you are using this tool right now. So currently, there are only few tools in the world that are industry strength, and people are using that for target discovery. So Pandomics, I think right now is probably the most popular in the pharmaceutical industry. So now a lot of pharma companies are using Pandomics. We get substantial revenue there. Chemistry 42 allows you to do multi-parameter optimization of the compounds that you want to take to the clinic. So it utilizes multiple generative engines that allow you to generate a small molecule with the desired properties for a specific target with the known structure or without the known structure, with a known ligand or without the known ligand. Currently, 9 out of the top 30 pharmaceutical companies are using this tool internally. And we are also working on a system called Insilico. It's a 6.5-year effort, which hopefully will allow us to predict phase 2 to phase 3 transitions utilizing predominantly preclinical data and also clinical study design data. So we are piloting with multiple hedge funds, banks, and if you like to collaborate on that, we are very open. So read up on our work, and we are very collaborative. Internally, we have a pipeline of 31 therapeutic programs. Of course, for legal reasons, I'm allowed to disclose only a few of them that have progressed. So one has progressed all the way into phase 1, and that's what I'm going to talk about. I'm here disclosing the targets. So, by the way, this is the target that BioAge is also going after. So, Christian, Godspeed. Considering the fact that we managed to identify the same target and prioritize it for aging, using 2 different approaches gives, a little bit more confidence that it's likely to work. And since 2021, we nominated 8 preclinical candidates. Preclinical candidates, it means that you have all the preclinical work ready for IND-enabling studies complete. Usually it's several preclinical models need to show in vivo efficacy. So we've got 8, so now we're racing. To my knowledge, that's a record in the industry at this point in time, so combining AI with the best superhumans and also massive CRO network where you can do massive parallel experiments. And in 2020, once we raised a little bit of money, nowadays we can call it a little bit, we decided to go after a grand challenge, which is trying to link biology and chemistry together in one go to identify novel targets for a broad disease with no cure, and targeting the fundamental mechanism of aging, because that's what I stand for as well. But of course, we need to ensure that it's in a classical pharmaceutical model, so we can later partner this program with pharmaceutical companies or progress it ourselves into the clinic, because currently, again, aging is a disease or not a disease. It's kind of Schrödinger's aging, depending on how you look at it and which paper you read. But regardless of whether it's a disease or not, it's a good idea to have a dual purpose target, which targets aging and disease at the same time. And we decided to go after fibrosis, or what I call xenofibrosis or fibrosenescence. And we decided to purpose our fibrosis work towards several therapeutic indications. One is IPF, idiopathic pulmonary fibrosis. It's an age-related disease, happens late in life, and it's pretty fatal. So people with IPF expire very quickly. Currently, there are only 2 approved drugs, nintedanib and pirfenidone. And even though the condition is reasonably rare, those 2 drugs drive over $3 billion in revenue annually, which makes it a pretty attractive, lucrative market, even if we're targeting this only a small disease. So this is probably the most important slide, because that shows the workflow and the thought process that we've undertook to reach this stage. So we first started with training a deep neural network that is trained to predict age, and then we retrained it on multiple types of fibrosis, on multiple tissues. That network is currently, we're using it internally. We did feature importance analysis, so looked at the most important genes, put them into biological context using our Panda-omics engine. You can get a demo of that, and actually there's going to be a presentation of Panda-omics if you are interested in making your targets commercializable. It's a good system to use because it also gives you some business intelligence. Then we assessed the novelty of the targets, because in target space, novelty and confidence go hand in hand. If the target is not novel, the pharmaceutical industry is not going to go after that. I mean, look at Tor, right? Beautiful target, many approved drugs, many amazing tool compounds, and for some strange reason, we don't see a commercial avalanche of clinical trials trying to progress TOR inhibitors into different therapeutic indications for age-related diseases. There are some. But if the target is too novel, if it's completely unknown, you have to do a lot of work on the biology side to validate the target, and the pharmaceutical industry, pharmaceutical companies usually will partner with you only after phase 2 is complete or at phase 2. So you will need to have courage and the resources to invest. So we prioritized novel targets, 20 novel targets. We managed to test only 5 because only for 5 we had more or less ready assays available. All of them worked, and they worked in vivo, and now I'm going to tell you the story. So we prioritized one of those targets that we consider to be the most important one, implicated it in lung fibrosis. We have several models, cells and mice. Skin fibrosis, currently we have in vitro efficacy, completing the in vivo efficacy package right now. Liver fibrosis and kidney fibrosis, we also nominated a preclinical candidate. So the beauty of what we do is we also do it with our own chemistry. So we don't in-license the compounds, or we don't take them from the molecular libraries. We generate them from scratch using Chemistry 42 tool. And then we synthesize and test. So for this program and for this target, we synthesized 79 compounds. Most of them were low double-digit nanomolar hits. Some were completely drug-like, and we did just two iterations of optimizations to get to 055, the 55th compound that we synthesized. And that's what we nominated for preclinical candidate. We also did many experiments. So here are just the highlights. We probably did about a dozen in vivo experiments. This is a lung function experiment. So this is a mouse. You put it in a very specific chamber where lung function is measured. This is the wild type, so no drug control. This is the bleomycin-induced fibrosis. So you've got lung function diminished. You can rescue it using a current standard of care, nintedanib, to some extent, at 60 mg per kg, so it's a very high dose. This is our compound at 60 mg per kg, so we slightly outperform, but at that level, it's actually pretty insignificant in terms of impact. But basically, you need to show comparable efficacy. But this is cool. So it has really nice dose response, and this is our 055 at 6 mg per kg. So at very low concentration, we still can rescue lung function after injury. This is a very important biomarker that most pharmaceutical companies want you to have in the data room if you are to talk about IPF. It's fibroblast to myoblast transition, FMT, and epithelial to mesenchymal transition, EMT. Basically, to cut the story short, this is nintedanib. This is R055. In FMT, we outperform 5-fold. And in EMT, we outperform 16-fold, which is pretty cool. We also show synergistic effect with pirfenidone. And of course, it's a completely different mechanism of action, so it's possible to do combos. So to complete the preclinical package, actually, you might want to take a picture of the slide just in case, because when I was getting into this field, I thought it was easy. So you need to complete multiple efficacy experiments, ADME studies, safety tolerability studies, do basic tox in mice. And then we nominated the preclinical candidate. So we did it in under 18 months, from novel target all the way to PCC. That's a pretty cool speed, and to my knowledge, that's a record. We did it also under $2.7 million just for IPF experiments. And we announced in February 24th, 2021. Then we also completed all the IND enabling package elements. So you need to also show that the compound is very, very safe before you start human clinical trials. So we did 14-day repeat dose study in mice and dogs. And then we also did the 28-day tox. It's not ongoing, it was completed. It's a little bit an older slide. So it turned out to be safe. And then we decided, since it's a novel target, we decided to do a small phase 0 study. So only 8 human healthy volunteers in Australia, and we completed it within a couple months, doing microdosing, and entered phase 1. So right now we are, so we entered phase 1 February 24th. It was supposed to be a big announcement, but Russia invaded Ukraine, and that was a terrible event for all of us. Deep bow to the citizens of Ukraine. So unfortunately, it happened, and of course we couldn't celebrate. But it was a pretty important milestone in our company, the start of phase 1. We did it in under 30 months. By the way, 24th is also my birthday, so I wouldn't be able to celebrate. And we also have another compound, 073. It works pretty much in the same way as 055. It's a little bit less clean. But we nominated the preclinical candidate for kidney fibrosis as well. So multiple efficacy models, in vivo efficacy. And again, to my knowledge, it's the first time when the drug was discovered using AI, so this novel target. Novel small molecule out of AI. We also did synthetic route planning using artificial intelligence, and then we went all the way into phase 1 human trials. So right now, fingers crossed, I'm writing on this program, but we also have 30 other programs, which is pretty cool. So since we've discovered that using an aging clock, I think it should work in aging. It's very difficult to make the claim at this point in time, but I think modulating fibrosis is a very important tool for studying aging. I know some scientists and physicians who think that aging is fibrosis, fibrosis is aging. So let's keep the fingers crossed. That's my contacts. We're actually soon opening in the Middle East and Montreal, and we're hiring, so shameless promotion. And you can follow me on Twitter personally, so I'm covering the conference. That's why I'm madly going around taking pictures of all of you. And I wanted to also announce a big thing in my life. So it's a longevity pledge. So as you know, I'm a single childless entrepreneur. So I will continue being single childless, hopefully, but maybe not. But I'm not planning to leave an inheritance, so I'll spend everything that I'm earning in longevity. So I participated personally in the last 2 rounds of Insilico medicine, and I'm investing in some of the companies around here. And I will be supporting some of the young scientists. We'll put more money into this conference personally next year and other conferences. So if you have some interesting event that you want to sponsor, I can give a little bit of a grant, but also I can provide substantial promotion support as well. And I will be trying to get more IT entrepreneurs into longevity, because that's how I switched in 2004, and I think it was a good thing. So all the details of this pledge are outlined on this website. It went live today, so I actually personally had to create it over the weekend. And thank you very much. Hope to collaborate with everybody. And next year is going to be our 10th annual. So it should be grand. Thank you.
Thank you so much, Alex. And I think we should also give him another round of applause for starting this whole initiative almost 10 years ago.
Thank you. Well, it's yours now.
we have any questions for Alex? I can start. So when do you think we are going to have one of your drugs available in the pharmacy?
Well, due to legal regulations and other regulatory regulations, I won't be able to speculate on that. But for a company of our type in that particular position, if everything were to go right, the phase 2 data would come in probably end of 2023, 2024 beginning, early. So that's when you know efficacy. And after that, it could be another 2 years. I keep my fingers crossed for Christine Fortney VIOH. I'm sorry. She is much further much further ahead because she in-licensed the drug. We had to get our own chemistry.
Right.
But we also have partnered molecules. So we support a lot of pharmaceutical companies. Recently, we partnered with Fosun Pharma on a really cool target that we disclosed called QPCTL. It's an Golgi complex, and it's in the CD47 pathway. We purposed it towards immuno-oncology. But actually, it has a role in aging as well. So we progressed it to preclinical candidate in February. Currently, it's an IND enabling. Hopefully, we'll be able to get into phase 1 early next year. Fingers crossed. That target actually might progress even faster.
All right, very nice. We have a question here in the front.
Oh, on one of your slides, I saw that you have MET2A as one of your targets. And I just want to mention, like, MET2A is a methionine cycle, and it's very hard to achieve.
It's a SAMI regulator, yeah.
Yes, so it is very hard to achieve methionine restriction via dietary interventions, almost impossible. So it would, I don't know if you have a small molecule to inhibit it, it could be a methionine restriction mimetic that everyone is wanted in the field.
Yeah, so as a matter of fact, so this target has a really cool story, just like USP1. USP1 is a really interesting target as well. It's the area of synthetic lethality, which is super hot in oncology. But MAT2A, so we actually do it for commercial purposes, right? So we're a commercial company, we need to make money. So those are hot targets. We also have a system which predicts how hot they're going to be in the future. So we bet on those two and progressed. So right now there's a lot of interest for cancer. But MAT2A, since it regulates SAMI and methylation, could be used as a tool compound, molecule drug maybe, to do all kinds of methylation experiments as well.
Very cool. We have another question here.
Hi, Alex. Thanks for your talk. I was wondering about one of the molecules that you were presenting. I think it was 055. You were measuring the lung function in the mice, and you had also mentioned that the drugs that are on the market improve FEV, but not oxygen saturation. So I was wondering if you're able to measure that in mice, or if you're worried that that might be a shortcoming of the drug that you're pursuing as well.
So at this point in time, we just completed the same experiments that the current standard of care, where there is published data on nintedanib. But of course, that's a really valuable advice to do an additional experiment on lung function. Currently, we see that we rescue fibrosis in the lung after injury by bleomycin, but also some other injuries as well. Currently, our approach is very, very standard that the biotech would take, so you need to do head-to-head comparison with current standard of care.
Right, nice. We have one from one of our Inspire ambassadors, Andrea.
Alex, it's not really a question, but rather it's a really, really big thank you from the whole RDD ambassadors team for helping us get here and for making this possible. And from myself as an Insilico medicine intern, it has been an incredible journey these 2 years interning at your company, so thank you.
Yeah, well, hopefully you'll publish a paper. That will be super cool.
All right, thank you so much, Alex.
Thank you.